It's my pleasure here to introduce Dr. Emily Hemann. Dr. Hemann is an expert virologist. She has extensive experience studying the immune response to influenza A virus, in addition to other viruses and infectious disease. She actually began her academic career at the University of Iowa, where she received her Ph.D. Her graduate work was carried out in the lab of Dr. Kevin Legee, where she studied the roles of pulmonary dendritic cells and Cd8 T cells in acquiring protection following both influenza A vaccination and infection. She also looked at important big picture questions like how extrinsic factors such as ethanol exposure or diet-induced obesity impact immune response. I thought that was really awesome. She went on to pursue a postdoc out here at the University of Washington in the lab of Dr. Ram Saban, where she studied the innate-adaptive immune interface during virus infection. It's here where she received too many grants and awards to list, including several from the NIH, the American Association of Immunology, and the International Cytokine and Interferon Society. One of her lesser known postdoc accomplishments was going to the local barbecue joint down here in South Lake Union, Cask and Trotter, and teaching others how to correctly eat chicken wings. She then moved to her current location at The Ohio State University, where she's an assistant professor in the Microbial Infection and Immunity Department. And she established her lab this past year. The Hemann Lab researches and focuses on understanding the contribution of interferon lambda alongside other cellular components and signaling pathways that function within the innate-adaptive immune interface to program protective immunity against viral infection. I'd like to close with a little fun fact about Emily. She once won a radio contest here in Seattle to meet the famous American rapper Flo Rida in person. I must say I'm a little bit jealous that she got to meet him. And I spent this morning listening to some of his old hits. So please join me in welcoming Dr. Emily Hemann.
Thank you very much for that introduction. I'm very excited to share my work with you today. So I'll be telling you about what we're working on, looking at the role of interferon lambda at the innate-adaptive immune interface and its roles during influenza virus infection. So we've been a lab here for just about 10 months, which in COVID times is really about 3 months of doing experiments. But I do have some new data that I'm really excited to show you all today, looking at this important role of interferon lambda during flu infection. So we may have forgotten about it at least a little bit in the last year, but influenza virus still remains a persistent global health burden, at least in years when we're not masking and social distancing. So I predict, as I'm sure many others in the field, that this is going to continue to be an issue moving forward. But just briefly, influenza virus is a negative sense single-stranded RNA virus whose genome is divided into 8 segments that encodes for 11 or 12 proteins. And influenza virus, while we think of it as being sort of a general nuisance, in the year seasonal flu season from 2019 to 2020, this led to about 50 million illnesses, about 26,000 hospital visits, 700,000 hospitalizations, and 50,000 deaths. And this is what occurs in what's thought to be sort of a general year. So this is a non-pandemic year. We typically don't think of influenza as being something that's that bad, most of us, but clearly it remains a global health burden, despite the fact that we have vaccines and antivirals against influenza virus. And one of the reasons for this is that influenza virus is unique, primarily because of this segmented nature of the genome. It's able to infect multiple virus strains, are able to infect a common cell, such as one within a pig. And this can lead to recombination of these viral gene segments known as antigenic shift, where you have full shifts of these viral genes, leading to the production and spread of rapidly divergent viruses for which the human population generally has no protection against. So for example, this is what happened in the case of the 2009 pandemic H1N1. And so one of the reasons that this can occur is due to the nature of our vaccinations. So one of the most commonly given vaccines is a split vaccine, where essentially virus is produced, detergent is used to split these proteins apart and uses a protein-based immunization. And so this vaccine does a really good job of inducing a hemagglutination inhibition response. So being able to, antibodies that are able to bind the virus, and as well as producing antibody secreting cells. But what the vaccine doesn't do is induce a robust nasal IgA response. So IgA in your nasal passages where you would likely be encountering a virus in the first place. It doesn't do a very good a job of inducing Cd8 T cells. So I'll tell you why that's important in a second. But overall, this leads to a lack of cross-protective immunity, which means that the strain that you're vaccinated against is very specific for that strain. So a lot of changes can occur in this virus that allows it to circumvent our vaccination-mediated protection. And so this is due to the fact that when you have vaccination for a target strain, right, you generate antibodies that bind to this very well. But when we have antigenic shift, which we just discussed, you can have massive changes in the viral protein structure that subvert antibody-mediated neutralization and protection. And additionally, this virus can mutate very rapidly, leading to changes in antibody binding sites through antigenic drift. And so both of these can undermine the durability of vaccination, and is one of the reasons why when we have emergence of a new strain, and from year to year, we generally need new vaccinations. And so one of the questions that we're interested in is how can we induce broadly protective adaptive immune responses following vaccination, not only necessarily with split vaccine, but with any vaccine platform. So we think that paying more attention to some perhaps more conserved types of immune responses may be important in developing better vaccines moving forward. And so we're really interested in Cd8 T cells. Why do we care about Cd8 T cells, right? Most of you have heard of it in one seminar or another. You study it probably in the context of cancer immunology, immunity in general, and there have been many studies in mouse models showing the importance of Cd8 T cells in protecting against flu infection. And it wasn't really until the 2009 pandemic that it was finally observed in a human population that Cd8 T cells do mediate protection. So at the outbreak of this pandemic, H1N1 seronegative individuals, so those who didn't have antibodies specific for this virus, it was determined whether or not they had T cells that were specific for conserved peptides between this virus and previously circulating viruses. And then individuals were monitored for clinical outcomes following pandemic H1N1 infection. And what they observed was that individuals who, again, had no antibodies for this virus, but had conserved T cell responses, had a lower flu symptom score. So they weren't as likely to become ill if they were exposed to the virus or if they became seroconverted. And so this really highlights the fact that in the human population, Cd8 T cells do really play a role in protecting against infection. And so just to make sure that we're all on the same page, typically in the respiratory epithelium, there are dendritic cells and antigen-presenting cells that are surveying the airways. When they come across a virion or a virus-infected cell, they're able to uptake that antigen. They can migrate to the lung-draining lymph node and present that antigen in the context of MHC to a naive T cell specific for that antigen. And here we're just showing you a Cd8 T cell. The Cd8 T cell will then rapidly proliferate after it becomes activated. It will upregulate effector functions and then migrate back to the lung environment where it's able to kill virally infected cells and lead to clearance of the virus and then establish long-term memory protection. And so while many studies have really been trying to dissect this over the years, what we think is really fascinating in the lab is that pretty much all of this is kicked off by these sort of initial viral sensing pathways in infected cells. And so the central question that we're trying to answer in the lab is how are virus recognition as well as interferon signals integrated to induce adaptive immune responses against infection, with the ultimate goal of trying to harness these pathways as therapeutics or adjuvants. So following influenza virus infection, It's sensed within the cell by TLRs or cytosolic Regi-like receptors, Regi and MDA5. These lead to activation of the downstream adapter MAVs and activation of the IRFs as well as NF-kappa B to lead to the production of interferon, cytokines, and antiviral genes. Interferon specifically is then secreted, and it can bind to the interferon receptor on the same cell or within neighboring cells, leading to a broad antiviral state within the tissue. And so we know, and it's been very well characterized, that interferon in general is induced in the lung following flu infection. So both interferon lambda or type 3 interferon, as well as interferon alpha beta, also known as type 1 interferon, are both induced following infection. However, there are some key differences between these two pathways, despite the fact that their canonical signaling cascades that had been originally described are relatively similar, leading to downstream activation of JAK-STAT pathways, as well as NF-kappa B, leading to the upregulation of interferon-stimulated genes. Interferon lambda, while it was originally described as an interferon given this activation of the JAK-STAT family, it actually is more structurally related to the IL-10 family of cytokines and signals through this heterodimeric complex of the interferon lambda receptor and IL-10R2. The expression of the interferon lambda receptor is very limited compared to type 1 interferon. So type 1 interferon is expressed in nearly all nucleated cells, whereas the interferon lambda receptor is expressed primarily on epithelial cells, human hepatocytes, neutrophils in mice, plasmacytoid dendritic cells, but we still don't really understand the full complex mix of cells that are able to respond to interferon lambda, primarily because we're still lacking a good antibody to look at this receptor. Nevertheless, we do know that it's relatively restricted compared to type 1 interferon. We also know that while the same signaling pathways are engaged, a number of comparative studies have demonstrated that interferon lambda induces a less inflammatory response compared to type 1 interferon. And so typically, most of us think about type 1 interferon being induced downstream of influenza virus infection, but it's actually interferon lambda that's the predominant interferon produced within the lung environment following IMV infection. Additionally, in studies where mice were infected with influenza virus and given interferon alpha as a therapeutic post-infection, this led to a reduction in virus. So it was able to knock down virus titers, but the highly inflammatory nature of this type 1 interferon actually enhanced pathology and overall reduced survival of these mice. However, when interferon lambda was administered as a therapeutic, it also reduced virus, but given the less inflammatory nature of interferon lambda, it also reduced pathology and enhanced survival. So not only is interferon lambda the predominant interferon induced following flu infection, it plays an important role and really can serve as a superior therapeutic to influenza virus infection compared to type 1 interferon. However, despite these roles in mediating protection, really there's not a whole lot known about the role of interferon lambda in mediating adaptive immune protection following flu infection. So a study from a couple of years ago now used an attenuated vaccine strain of influenza virus and administered it intranasally with interferon lambda. And what they observed was an increase in IgG and IgA within the bronchial alveolar lavage fluid of these mice, demonstrating that interferon lambda can actually augment the production of antibodies within the airway space following an intranasal vaccination. However, the contribution of interferon lambda to regulating Cd8 T cell responses during flu infection has remained relatively unknown. So it was at the start of this project, there wasn't any published work really demonstrating whether or not interferon lambda played a role in contributing to Cd8 T cell immunity to influenza virus infection. And so we predicted that interferon lambda was important in regulating the Cd8 T cell response at the innate-adaptive interface. And so we had both wild type as well as interferon lambda receptor knockout mice available to us to be able to start to answer this question. So the first thing we did was we infected mice with a sublethal dose of H1N1, and 9 days later we harvested the lungs. We then took these lung homogenates and stimulated them with the conserved influenza virus specific peptide, NP366, and looked at cytokine production, both TNF alpha and interferon gamma. And so what you can see here is when we stimulate these lung homogenates with NP366, there's a significant reduction in the amount of cytokine produced by these Cd8 T cells. This really suggests to us that interferon lambda is playing an important role in supporting the full capacity of this Cd8 T cell response. When we look over time, this correlates to an increase in virus titer. So you can see here on day 5 and day 7 post-infection that there's a significant increase in interferon lambda receptor knockout mice following flu infection. And what we also see over time is a delay in clearance. And those of you in the crowd who are probably looking at this and virologists, and you're saying this is only a half log increase, what does this matter? You're right. For the context of a primary infection alone, it's really not that important. But as I mentioned to you earlier, we know that the CD8 T cell response plays an important role in mediating protection in situations where you don't have pre-existing antibody responses. So we wanted to see whether or not these CD8 T cell responses that were significantly reduced at the effector stage, does this translate into a loss of protection in a model where we know that T cells are responsible for mediating protection against flu? And so we can actually model this in our mirroring system. So if we administer H1N1 to mice, we generate neutralizing antibody responses as well as flu-specific CD8 T cell responses. And if we come back and infect these mice 30 plus days later with the same virus strain, these neutralizing antibodies are pretty much able to blunt infection from the get-go, but you do have these flu-specific CD8 T cell responses that are able to clear any remaining infection. And these mice really don't tend to get sick at all. However, we can use a recombinant H3N2 strain where the hemagglutinin and neuraminidase, so these two surface molecules, are from an H3N2 strain. And all the other gene segments are from this same H1N1. And so in the context of this virus infection, we again develop neutralizing antibody responses as well as flu-specific CD8 T cells. When we come back again and give a high dose of this H1N1 strain, in this case, it's been well documented by numerous studies over the years that these antibodies are no longer able to neutralize, and it's really conserved T cell responses that are required for mediating protection. And so again, to see if interferon lambda signaling is required for T cell mediated memory protection, we took our wild type or interferon lambda receptor knockout mice, administered a sublethal dose of the H1N1 or H3N2, and 45 days later, we administered a high dose of either of this H1N1 and monitored for morbidity and mortality. And so in this case, we can look at the contribution of both antibodies and T cells, or just T cell mediated protection in this rechallenge system. And so showing our controls here, if we have mock animals, they all go on to do just fine, as you would expect. Whether we have wild type or knockout, if they only receive this high dose secondary challenge, they succumb to infection with similar kinetics. If we administer wild type or interferon lambda receptor knockout animals H1N1 followed by H1N1, so we have both neutralizing antibody as well as T cell mediated protection, we don't see a significant reduction in survival in either of these groups. And when we've gone on to look at antibody responses, we don't see any defects in antibodies in either the serum or the bowel of these animals. When we administer H3N2 followed by H1N1, as it's been previously established in the literature, we don't see a significant reduction in survival. However, when we infect our interferon lambda receptor knockouts with H3N2 followed by H1N1, we now see an approximately 50% significant reduction in survival, suggesting that interferon lambda is important for T cell mediated memory protection against influenza virus infection. And so one of the things that we're looking at now and we're really interested in is trying to determine what type of memory T cells are these that are mediating protection. And is interferon lambda important for developing both central and tissue resident memory T cells? So central memory T cells are CD8 T cells that generally circulate between lymph nodes. They have high proliferative capacity. And compared to tissue resident memory T cells, these typically reside in the lung following influenza virus infection and are thought to rapidly upregulate effector functions to mediate protection. And what's important is that both the central memory as well as tissue resident memory have been shown to mediate protection in murine models of influenza virus infection. So if you've been reading the flu literature in the last few years, there's been a lot of focus on these tissue resident memory cells, and they do play an important role, but these central memories can also be sufficient to provide protection. And so to start to look at this question, we infected mice with our wild type or interferon lambda receptor knockouts, again, with a sublethal dose of H1N1. I'm going to show you our data from 15 days, but we've also looked at day 30 and see very similar results, and then harvested the lungs and the draining lymph nodes. And so first, I'll just show you the draining lymph node data. So I'm showing you data from the same experiment that I'm going to show you next, but we have already published data from the lymph node previously demonstrating that at the time of rechallenge, so day 35 post-infection, what we observe is a population of flu-specific CD8 T cells that are present in the draining lymph nodes of wild-type animals, and that this is significantly reduced in the interferon lambda receptor knockouts. So in this case, again, we have 4 animals divided into 2 time points, which we can't do statistics on, but I think it's important to show you data from the same experiment that I'm going to show you next. So within the same experiment, if we now look at the development of tissue resident memory cells, we do this by administering CD45.2 intravenously 3 minutes prior to harvest. And so by doing this, what we can do is label all of our CD45.2 positive cells in the vasculature, but the tissue niche remains protected, and we can delineate what's in the tissue versus what's circulating in the vasculature. And so just to show you this is a proof of concept for the IV labeling, if we look at our mock animals, you can see that many of the CD8 T cells that are present are in the vasculature. So they're IV positive, they were stained in that 3 minutes. But when we look at day 15 following influenza virus infection, what you see now is that the majority that remain are in this IV negative fraction. So they are within the tissue niche themselves. And what's really interesting, and I think an unexpected result for us, is that when we looked at these flu-specific CD8 T cells by tetramer staining, what we observed was that there was no difference in the percentage or numbers of these flu-specific CD8 T cells that were in the lung tissue at day 15 or day 30 post-infection. And when we looked at CD69 and CD103, hallmarks of these tissue resident memory cells, and these molecules are important for keeping them within the tissue, we also don't see differences in the percentage or distribution of these molecules. And so this really suggests to us that interferon lambda seems to be important for generating a memory population within the lymph node, but it doesn't seem to be required for TRMs. And we think this is really curious because I showed you previously that at day 9 post-infection, we did see a significant reduction in our flu-specific CD8 T cells within the lung. So some of the things we're doing now to really start to look at this in depth is to see whether or not there are functional differences in these cells that are still remaining in the lung that seem to get seeded to similar levels, so this tissue residence data at day 15 and day 30, whether or not they persist similarly, but also really going back and looking at the central memory population and seeing whether or not they have defects to proliferate and whether or not they could really be the ones that are defective in mediating protection. And so to briefly summarize this first part, looking at the role of CD8 T cells specifically. So following influenza virus infection in the absence of interferon lambda signaling, we see a reduction in CD8 T cell proliferation, reduced flu-specific CD8 T cell populations at an effector time point. This leads to an increase in virus titers and reduced T cell dependent memory protection. And again, what's really intriguing is that we see a significant reduction in our T cell populations within the lymph node, but strictly looking at numbers of tissue resident memory cells within the lung, we don't see a defect, suggesting that perhaps there are different roles being played in the lymph node versus the lung, but we're still looking into this. So what we next wanted to know is whether or not this interferon lambda mediated defect is CD8 T cell intrinsic. And so to look at this, what we did was we took wild type or interferon lambda receptor knockout mice, and we transferred in CFSC labeled P14 T cells. So these are transgenic T cells that are all specific for this GP33 epitope, and infected them with a virus expressing this GP33 epitope that we received from Ryan Langla at the University of Minnesota. And on day 3.5 post-infection, we harvested the lung draining lymph node to look for activation of these cells by proliferation and CFSC dilution. And so in our wild type setting, so all of these cells are GP33 specific, they're all wild type, but when we transfer them into wild type hosts, you can see that the CFSC is pretty much diluted out, but this is significantly blunted in our interferon lambda receptor knockouts. And if we determine the proliferation index, so the average number of divisions that the cell population as a whole has undergone, this is significantly reduced in the interferon lambda receptor knockout, suggesting that this is not a CD8 T cell intrinsic defect that we're seeing, but rather an intrinsic one, as these CD8 T cells, again, are all of the same specificity. We've also done additional ex vivo experiments and again see that there's really no intrinsic defect in CD8 T cells to be able to respond to influenza virus infection or be activated in the absence of interferon lambda signaling, that it's really something extrinsic to the T cells themselves. And so because this defect we saw was as early as activation within the draining lymph node, we next hypothesized that interferon lambda programs dendritic cells to facilitate their function and regulation of adaptive immunity. So it's also possible that epithelial cells are playing a role in somehow programming this state. This is something we're looking into as well, as epithelial cells are known to respond to interferon lambda. But again, given that the defects we were seeing in the T cell response were happening very early on, day 3.5 post-infection, we thought it would be really interesting if perhaps interferon lambda was regulating dendritic cell function. And so to start to look at this in sort of a higher level in a model of infection, we took interferon lambda receptor floxed mice generated by Megan Baldridge and Skip Burgin, kindly shared with us. We crossed them to CD11C Cree or Lysem Cree animals. So in this model where we cross them to the CD11C Crees, this would ablate the interferon lambda receptor in dendritic cells, macrophages, and neutrophils. And we also, as a control, cross them to Lysem Crees. And so this would ablate interferon lambda receptor in macrophages and neutrophils. And we thought this control is important. So while the role of interferon lambda signaling in macrophages is still unknown, it is known that interferon lambda signaling plays an important role in neutrophils in regulating host immunity to influenza virus infection. And so by comparing these two animals, really the difference in these crosses is that in this cross, interferon lambda receptor by CD11C, we're knocking out the same cell subsets, but just dendritic cells in addition. And so when we look in our interferon lambda receptor floxed mice compared to our floxed by CD11C Cree, we see a not complete but significant reduction in our number of flu-specific CD8 T cells that are present in the lungs on day 9 post-infection. And when we compare this to our Lysem Cree, again, in this case, we're only ablating the interferon lambda receptor in macrophages and neutrophils, we don't see this significant difference. So this really suggested to us that it was interferon lambda signaling in dendritic cells that was required for generation of a full magnitude CD8 T cell response following infection. And like I mentioned earlier, we can't just do immunohistochemical staining to look for the distribution of the interferon lambda receptor, because currently there's still no antibody that specifically stains for this receptor on cells. And so we went to our BMDC model to be able to look at this and determine whether or not they were able to respond to interferon lambda. So first, to see whether or not it was expressed at least at the message level, what we noted was that there's detectable message in our wild type bone marrow derived dendritic cells. And when we infect with influenza virus, the interferon lambda receptor message is actually increased. This is compared to our interferon lambda receptor knockout with just how this deletion is made. The mRNA is unstable and undetectable. So these BMDCs do seem to express levels of the interferon lambda receptor, and actually they're able to upregulate that message upon infection, which is quite interesting because we have also shown that type 1 interferon receptor, as well as IL-10R2, the other component of this receptor subunit complex, remains stable over the course of infection. So the ability to upregulate this interferon lambda receptor in dendritic cells seems to be a novel feature of the interferon lambda receptor, perhaps particularly in immune cells or maybe dendritic cells. But we then went on to stimulate these cells with either interferon lambda or interferon alpha and look for activation of the downstream JAK-STAT signaling pathway by phosphorylation of STAT1. And so these cells are indeed able to respond to interferon lambda as well as type 1 interferon. And by using BMDCs from either type 1 interferon receptor knockouts or the interferon lambda receptor knockouts, we confirm that this is a specific activation and it's not due to crosstalk between these two pathways. So interferon lambda is able to fully signal in the type 1 interferon receptor knockout animals, but not in the interferon lambda receptor knockouts. And the same is true for type 1 interferon. So these cytokines are both able to signal in our BMDCs through their specific pathways. We next wanted to know whether interferon lambda signaling was required for DC function. And so we know that for activation of Cd8 T cells, they need to be presented antigen in the context of MHC, co-stimulatory molecules, as well as cytokines. And so to start to look at this, we treated dendritic cells with DQ-OVA. So this is a molecule that when it is taken up by the cells and cleaved in the endosome, as it turns into the lysosome, it becomes fluorescent. And so you're able to measure antigen uptake and processing by an increase in fluorescence within your cells. And so when we treated BMDCs with this DQ-OVA molecule, what we observed was at not only a steady state, but also throughout the course of infection, these dendritic cells generated in the absence of interferon lambda signaling are unable to upregulate or increase this signal over time, demonstrating that interferon lambda signaling is important for antigen processing and presentation. Additionally, and this is published, we looked at co-stimulatory molecule upregulation, and this is blunted in flu infection in vitro in our BMDCs lacking interferon lambda signaling. So we next wanted to go back in vivo and see whether or not there was a difference in DC subsets that were being recruited to the lymph node of our interferon lambda receptor knockouts following flu infection. So we know that following influenza virus infection, multiple cells are going to migrate to the draining lymph node, but we know in the context of flu that CD103 positive dendritic cells play an important role in carrying antigen from the lung to the draining lymph node to then hand them off and cross-present antigen to T cells. We also looked as a proxy at CD11b positive inflammatory dendritic cells because these are also known to migrate. But by and large, we were just looking to see whether or not there was a change in distribution of these dendritic cell subsets within the lymph node, because again, we saw this defect in the T cell response as early as 3.5 days post-infection. And so looking here at our CD103 positive dendritic cells, well, these are accumulating in our lymph nodes of wild type animals at day 4 post-infection. This is significantly reduced in our interferon lambda receptor knockout mice. And interestingly, when we compared this to our inflammatory dendritic cells, which are also known to migrate from the lung to the draining lymph node during infection, there's a bit more spread here, but we do see an increase in accumulation of these inflammatory DCs, and there's no significant change in the absence of interferon lambda signaling. So this accumulation of CD103 DCs specifically in the draining lymph node seems to be occurring in the absence of interferon lambda signaling. And we think this is very interesting because previous work from Tom Moran's group has shown that CD103 positive DCs specifically are resistant to type 1 interferon signaling. So we think that the interferon lambda receptor and interferon lambda signaling could be playing an important and unique role in this dendritic cell subset that's been shown to not be as responsive to type 1 interferon and maybe playing an important role in activating these cells following infection or antigen uptake. And so just to demonstrate this in preliminary data, when we sorted these cells, these either inflammatory dendritic cells or CD103 positive dendritic cells, and looked at expression of either the type 1 interferon receptor or the interferon lambda receptor, what we observed was that there is a trend towards a reduction in type 1 interferon receptor. So in support of what's been previously published by Moran's group, demonstrating that these DCs are more resistant to type 1 interferon signaling. And we only detect this interferon lambda receptor message in our CD103 positive subset, again, suggesting that these CD103 DCs may be reliant on type 3 interferon to execute important activation and antigen presentation functions. And so because these CDCs seem to require the interferon lambda receptor and interferon lambda signaling for optimal function, we wanted to better understand how this signaling could regulate CDC programming during influenza virus infection. And so we performed an RNA sequencing experiment where we infected wild type or interferon lambda receptor knockout mice with a sublethal dose of influenza virus infection. 4 days later, we harvested the mediastinal lymph nodes or lung draining lymph nodes. We sorted out our conventional dendritic cells, which are largely made up of the CD103 positive DCs, with a few included Cd8 alpha positive CD103 negatives, and then performed mRNA sequencing. And what's clear here from the beginning is that there are a number of overlapping genes, but far more that are unique between these two subsets within the draining lymph nodes from the wild type or the interferon lambda receptor knockout, demonstrating that interferon lambda signaling is playing an important role in programming the gene signature within these DC subsets. And what's interesting is when we plot all of these genes in just a dot plot where we can see our wild type genes infected versus mock. So moving here on the x-axis to the right, these are genes that are being upregulated in infection or downregulated during infection in the wild type setting. And on the y-axis, we have our interferon lambda receptor knockouts. So upregulated on this y-axis moving up or downregulated in this y-axis moving down. We can see here a number of ISGs. And intriguingly, we don't really tend to see a lot of changes in ISGs that are happening within these two subsets. So it seems like something is occurring outside of sort of traditional interferon signaling or ISG activation, which again is probably not that surprising, given that Tom Moran's group has shown that these cell subsets really don't rely on type 1 interferon to a large extent to execute their functions in antigen presentation. But what did stand out to us was this gene over here that was very highly upregulated in the interferon lambda receptor knockout setting during flu infection, but downregulated in our wild type. And this gene was IL-10. So we also did de novo mapping to start to see which networks were being uniquely regulated in the interferon lambda receptor knockouts. And intriguingly, only one non-canonical interacting network fell out, and this included IL-10. And so compared to our wild type setting, this network is largely silent. So it seems as though interferon lambda receptor knockout CDCs activate an IL-10 immunoregulatory network. And at the center of this network, you can see here some REL genes that are associated with NF-kappa B signaling. And so what this really suggests to us is that interferon lambda signaling is playing an important role in allowing for appropriate activation of these conventional dendritic cells and not allowing a default immunosuppressive signature to take over and potentially blunt activation of our T cells. So we then went on to look at this in our BMDC model to try to validate these results. And when we infected BMDCs and took supernatants following infection, we were only able to detect IL-10 in our interferon lambda receptor knockout BMDCs, validating our RNA sequencing finding. And we think this is really interesting because it's been demonstrated by Martin Richter's group that IL-10 produced by antigen-presenting cells can directly reduce CD8 T cell antigen sensitivity and activation. And so one of the things we're looking at now and trying to figure out really is how this axis is really operating. So how is interferon lambda signaling normally in a wild type setting, leading to suppression of IL-10, allowing for full effector activation? And in the absence of interferon lambda signaling, how then are we getting production of IL-10 themselves? And we think that this is important because it could yield not only insights in our system of influenza virus infection, but given this important access for interferon lambda in really regulating effector versus immunomodulatory functions of DCs important for programming CD8 T cells, we think this could have broad implications for many settings, such as other infections or other disease states where we know CD8 T cells are important for mediating protection. And so to start to look at this role of IL-10 in regulating infection, what we did was we took either wild type BMDCs and infected them along with our interferon lambda receptor knockouts, or we took our wild type BMDCs and infected them, but added IL-10 into these cultures with the prediction that if IL-10 was the only thing being upregulated in our interferon lambda receptor knockouts that was inhibiting function, we would see a phenocopy of our knockout. And I'm just showing you CD40 here, but we've looked also at in vitro co-culture systems and activation of CD8 T cells, as well as in other parameters, so upregulation of genes. And what we see is that what we really get is an intermediate phenotype. So IL-10 does seem to be playing an important role in this phenotype that we're observing as far as the reduction in DC functions. But it seems as though interferon lambda signaling is playing an additional and important role in modulating function, as the addition of IL-10 alone doesn't seem to lead to a complete phenocopy of this signal here. And what I'm showing you here is just one dose, but we've also done kinetic experiments, and this is basically the maximal amount of reduction we get. So it's somewhere in the neighborhood of 50% in the parameters that we've looked at, as well as in CD8 T cell activation. So it does really seem as though IL-10 is playing an important role, and we may be able to harness that to understand what's happening, but there are also other defects in these DCs as well. And so to summarize the second part, looking at the dendritic cell response. So in addition to this reduction in the T cell response, leading to enhanced virus titers and this reduction in T-dependent memory protection, we also know that we see a reduction in DC function and migration in the absence of interferon lambda signaling. This seems to be specific to our CD103 positive dendritic cell subset, and that in the absence of interferon lambda signaling within these DCs, we see the activation of an IL-10 immunoregulatory network that is at least partially responsible for the defects that we're seeing in the ability of these dendritic cells to become fully matured and activate CD8 T cells. And so again, we're really trying to understand how this network is being regulated in the DCs, understand what other defects there are, because again, we think that there could be broader implications for this, in that if these dendritic cells are really harnessing interferon lambda for CD8 T cell activation, we think that this could be playing an important role in more settings than just influenza virus infection. And so that's something we're just starting to look at now, is the role of interferon lambda in other contexts in modulating CD8 T cell immunity. And so with that, I would like to thank the members of the lab, Octavia Hogebaum, who helped in generating the preliminary data from our lab that I showed you, Kyok Gong, our lab manager. At the University of Washington, Michael Gale Jr., my former mentor, as well as Ram Sabin, my de facto day-to-day mentor, who provided a lot of support and expertise in the Interferon Lambda front. Skip Virgin, previously at WashU, as well as Megan Baldridge for providing us with mice and insight on experiments. Brian Langlois for providing viruses, as well as Adriana Ferrero, another assistant professor here at Ohio State University, who's been helping us out with the RNA-seq analysis. I'd like to thank also, so we previously, a lot of this work is supported by an American Heart Association postdoc fellowship, and we're now supported by not only startup funds, but a K-22 from NIAID. So thank you for your attention. And with that, I will gladly take any questions.
Thanks, Emily. It doesn't seem like there's anything in the chat yet. Let's see. But I can go ahead and ask a question for starters. So you mentioned a number of things that got downregulated when in the knockout, your interferon lambda receptor knockout strains as opposed to your wild type, things like your co-stimulatory molecules. You also saw fewer CD107(3) positive dendritic cells. And I'm wondering if you've looked at, do those go up at all when you add additional interferon lambda to the system? Because you'd mentioned earlier that when you gave mice both the virus and lambda, they did better. So I'm wondering whether you get an increase in some of those things when you add even more lambda than you get with just endogenous infection.
Yeah, so that is actually an experiment that we propose to do in the grant that we have, but haven't been able to do it yet. So we want to see if interferon lambda is able to augment responses and augment Cd8 T cell responses specifically. So we plan to look in the context of infection, a live attenuated vaccine given intranasally, as well as in conjunction with a split subunit vaccine given intramuscularly. With the prediction that it could potentially work in all of those zones, right? If it's really executed, interferon lambda's function in modulating this is executed just through the dendritic cell population, these CD103 positive DCs. But we could see enhanced augmentation potentially via intranasal routes, given that epithelial cells could also be playing a role. Or it could be that there's something unique about the CD103 positive DCs in the lung compared to other tissue sites that are responding to interferon lambda as well. But yeah, we have plans to look at all of those things. It's a great question. We just have not done them yet.
Awesome, thanks. If anyone has questions, they don't necessarily need to put them in the chat. They can go ahead and unmute themselves. Okay, well, if no one else has any questions, I know people spend a lot of time on Zoom and Teams and various versions of that. I think we can give reactions. We can give rounds of applause. So let's go ahead and give Dr. Hemann a round of applause. And thank you so much for giving us a wonderful and very interesting talk on the interaction between adaptive immunity and innate immunity. I found it certainly quite interesting.
Yeah, thank you. And if anyone has any other questions that come up that you're interested in discussing further, want to ask, just feel free to reach out and email me. I'd be happy to chat more.